Hello, my name is Ziliang, and today I'm going to tell a story about peanut nodulation symbiosis. The modern agriculture heavily relies on the synthetic nitrogen fertilizer, which threatens the sustainability of global food production and degrades the environment. Legume establish root nodule symbiosis with rhizobia to get the nitrogen they need. Understanding the symbiosis mechanism will bring us further to a sustainable agriculture. Most legumes, including alfalfa, use a root hair entry to establish the symbiosis, while some plants such as peanut uses crack entry. The crack entry is more primitive, but it's understudied. Understanding this mechanism can improve the nitrogen fixation efficiency in peanut and facilitate the engineering of root nodule symbiosis in non-legume crops. Our hypothesis is that the whole transcriptome sequencing and whole genome bisulfite sequencing on roots after early rhizobial inoculation can capture the key genes and methylation controls of crack entry. And we hope to profile the dynamic transcriptome landscape and their epigenetic controls of peanut crack entry. To do that, we use a pair of sister inbreed lines, E6 and E7. A Rhizobial strain LBA was used for the inoculation, while the water treatment was used as control. The root tissues at 2 hours post-inoculation and 12 hours post-inoculation were collected for the RNA and DNA extraction. They were then submitted for the sequencing. I analyzed the long and short RNAs separately to identify the protein coding genes, the long non-coding genes, and microRNAs. Differential expression analyses were used to compare genes between treatment and control. The weighted gene network analyses were used to identify the co-expression networks for all RNAs. Differentially methylated regions were also identified and associated with gene expressions. The whole protocol can be seen in Figure 3. As a result, around 188,000 transcripts were assembled, including over 10,000 lncRNAs, 1,739 differentially expressed genes, and 235 differentially expressed lncRNAs were identified. This shows that peanut had a significant response to rhizobia at the early stage. 237 microRNAs were identified in our data, but only 26 of them are differentially expressed. This shows that the microRNA regulation might not be the key factor for the rhizobia response. The transcriptome expression also showed that the E6 and the E7 may have a different response to early infection. As we can see, there's a little overlap of DEGs between the two genotypes. The enrichment analysis of DEGs showed that the defense-related genes were enriched, as you can see in Figure 4. Indicating that symbiotic rhizobia also triggered the plant defense. Around 5,000 DMRs detected, but only around 400 of them are associated with the DEGs. Most of them are CHS sites, which shows that the gene expression is mostly controlled by the CHH methylation. From the Figure 5, you can have a view of the landscape of peanut transcriptome and methylation. The network analyzed in the middle showed that there was a complex network between the coding genes and the non-coding RNAs that might play a role in crack entry. In conclusion, our study provided the peanut whole transcriptome and the methylome response to early rhizobial infection. There were many regions with methylation change after inoculation, but most of them are not directly associated with a gene expression. In the future, I continue to mining the data, associated DMRs with non-coding RNAs. I also search the candidate gene that's unique to the rhizobia crack entry infection, and conduct the expression and function validation experiment. Thank you.